Right, we are now live on YouTube for your information, Walun. Right, so
all right.
Okay, I will do the recording. Okay, so it's 11:05. Alrighty. Ladies and gentlemen, good morning. Welcome to the February 2022 edition of the Malaysian Mangrove Research Alliance and Network monthly discourse. And for February 2022, we are so grateful, we are so thankful, and we are so glad to have Dr. Ng Wai Lun, an assistant professor at Xiamen University, Malaysia, specifically at China ASEAN College of Marine Sciences. So he's Wai Lun, I think you've been with Xiamen University since 2012. Is that a correct guess?
2018.
Oh, 2018. All right.
Yep,
so yeah,
fairly recent.
Okay. So 2018 you joined Xiamen University, but before that you've experienced maybe postdoctoral experience. Maybe you can tell us about your experience prior to joining Xiamen University. But I know I've been following your publications. Actually, started in maybe I think the one of the best one was your first article on the hybrids of mangrove hybrids in Malaysia. I think it was 2012 you were talking about.
Yeah, around that.
Right?
Yeah.
Yeah.
Yes.
That's right. So we want to hear more about that. And of course, you are always my reference actually for, especially when we talk about the botany aspect of mangrove plants, especially on the hybrids, which we agree is a topic less discussed, not only in Malaysia, in fact, throughout our mangrove communities globally. This topic is always in discussion, always open for arguments, and of course it needs more understanding. First of all, maybe why hybridization happens in our mangrove. Perhaps it's due to the high number of species diversity, perhaps, or maybe the pressure that they face due to a lot of anthropogenic impact, or perhaps now due to climate pressure, changes in our environment, pollution and whatnot. So today we will hear from Dr. Ng Wai Lun on the evolutionary genetics of mangroves. So basically the history, the dynamics, the changes happening at species and genus level, and hopefully to look at that from the management perspective, because we need to know, we are slightly different from our friends at Atlantic East Pacific, where in Indo-West Pacific we have high amount of plant diversity, as well as faunal diversity, as well as environmental characteristics or environmental parameters that affect the sustainability or sustenance of mangroves, and effects on mangrove forests would have effect on communities that depend on these habitats and ecosystem. That means all of us. So without further ado, we will have Dr. Ng Wai Lun addressing his lecture in the next maybe 45-50 minutes. And after that, afterward, we will have a chance, all of you will have the chance to pose some questions. You can do that by putting in or typing in your questions in the chat box, or maybe you can raise your hand afterward to directly ask question to Dr. Ng Wai Lun. And this is not a one-way traffic. You all are also invited to actually provide some suggestions, comments, or observations to actually increase our understanding together, as well as this would be a good opportunity also to create a new bond, a new friendship and network, as well as to maybe collaborate. Dr. Ng Wai Lun is also seeking for other experts to work together. And this also is a good opportunity for students, especially to maybe sign up for maybe graduate research with Dr. Ng Wai Loon and all of us in the MyMangrove community. So without further ado, Dr. Ng Wai Loon, the floor or the screen is yours. Yeah.
Thank you very much, Dr. Audrey. First, let me crack this screen sharing thing first.
Sure.
All right. Do you all see my screen? My slides over here?
You can now, yeah. Yeah, while you're preparing
your... Yeah, I can see it. Yeah, I can see it. Fong here.
So I would like also... I would like also, before Dr. Wai Loon proceed with the talk, I actually would like to thank all of you for being here, for participating in our discourse this month through Zoom and also through YouTube. Especially thanks to all of you all around Malaysia. I can see some very familiar names from Noaini, from Dato' Noaini from UMT, and then all our friends from other universities as well. And we have Stephanie all the way. I can't remember where Stephanie is based. I think she's in Australia or in the UK. Thanks for your time, for joining us. And yes, Prof. John Yong, all the way from Sweden. Thank you for joining us. All right, so let's hear from Wai Loon, and then later we will have a chance to discuss. Please, Wai Loon.
All right, thank you very much, Audrey. I have to say I'm very humbled, and I'm very grateful for Audrey to actually, congregate everyone to actually come for this MyMangrove monthly discourse. And I'm humbled by the attendance of so many VIPs, as well as colleagues in the arena of mangrove research. So a bit about my background. I obtained my bachelor's degree from University Tunku Abdul Rahman, so locally. And then after that, I did my master's and PhD in Kyushu University in southern Japan. All right, it was during that time that I was actually exposed to this mangrove research, and thankfully, by luck, probably, we stumbled upon hybrids, and part of my PhD study was on the hybrids as well. And after my PhD, I actually returned to Malaysia. I was a postdoc in UPM for, I believe, 2.5 years before I left Malaysia again for another postdoc stint in Sun Yat-sen University in China. And finally, when I got a position in Xiamen University, Malaysia, I returned hopefully for good, all right, in 2018. So evolutionary genetics has always been sort of like an interest of mine. So in evolutionary genetics, it's based on the very basic theories of genetics that has been established a very long time ago. However, what we see nowadays, it's more of technological advancement, all right? So using theories that were developed many years ago, with the advancement of technology, we actually advance in terms of evolutionary understanding. So my topic for today is evolutionary genetics of mangroves towards effective conservation and management. I believe a lot of the mangrove rehabilitation work nowadays depends on field biology as well as planting different types of techniques. But I feel genetics is very less heard of when it comes to the planning of rehabilitation or replanting, which I hope after this talk it will change a bit or improve a bit on how we actually handle rehabilitation as well as reforestation works. All right, so I'll start with the topic. This is my outline over here. I'll start with a basic introduction. Many of you would have already understood some background on mangroves, but I'll try to run through some background. And then I'll go through some fundamentals of evolutionary genetics. So I start with very basic genetics terminology. And then after that, we'll look at how evolutionary genetics actually work. Then we'll go into research on mangrove evolutionary genetics. I'll basically be using my PhD work, very briefly introducing what I've done for my PhD work and how we can actually utilize those information, okay, for mangroves. And then we'll look at what is the trend in terms of other mangrove species as well. All right, so we'll be looking at 3 subtopics over here. We'll be looking at genetic diversity. How is it important? How can we actually interpret that? All right, we'll also be looking at population genetic structure, what this actually means. And I believe it's actually very relevant when it comes to especially replanting, all right, and how we can actually look at that. And finally, introducing the topic of natural hybridization in mangroves, which like Dr. Aldri mentioned earlier on, is a topic that is less discussed in the mangrove research arena. And I believe it's important that we actually come together to talk about this because they are rare, and how we actually want to look at natural hybrids in terms of when mangroves are concerned. All right. And finally, I'll have just a 1-slider on what I think, okay, all these evolutionary genetic parameters can actually advise conservation and management. All right. So very quickly, mangroves, for those of you who are not familiar, the word mangroves usually can refer to the plant species itself. We can usually say mangrove species, or we can also say mangroves as in the habitat that these plant species actually live in, all right, the intertidal region that contains these plant species. So the common characteristics of mangroves usually include salt tolerance. That's very important because they usually grow by the intertidal region and therefore receive daily flooding or inundation of salt water, all right? And because of that also, and also the environment that they live in, muddy, sandy, submerged substrate, they usually have specialized root systems for basically 2 very important functions, for breathing as well as for support, okay? Because remember, the substrate over there is soft and fluid, and therefore they need to support their big body structures, all right? And finally, we also have the condition or situation of vivipary, whereby some of the seeds actually germinate while still attached to the mother plant. But of course, this does not happen in all mangrove species, but a significant number of them, okay? When it comes to mangrove diversity, that depending on the book or authors that you follow, all right, there are around, say, 60 to 80 species of true mangroves worldwide. So true mangrove species worldwide, okay, and most of them occur in the IWP region, which is the Indo-West Pacific region over here. So we also have the Atlantic East Pacific region with mangroves with rather similar coverage, but in terms of the species diversity, the Atlantic East Pacific is slightly lacking behind the Indo-West Pacific. When I say true mangrove species, they are the mangrove species that occur only in the mangrove environment. We also have other mangrove species that are called mangrove associates, all right? So over here, I just highlighted 2 types of mangrove associates that probably you will see very often in Malaysian beaches or mangrove environments, all right? We have the hibiscus tiliaceus as well as the ipomea. So this crawling plant over there, usually found along the beaches as well. And in terms of mangrove guidebooks, I find this recent publication by the Wetlands International Malaysia rather user-friendly. Okay, so whenever I bring students out for fieldwork to look at mangroves, I usually will share this book with them so that they can do some layman identification in the field as well. According to this book, there are 39 true mangrove species in Malaysia, all right, as well as 65 mangrove associate species. This is quite a lot, and I realized that some terrestrial species that we can also find in the forest are also listed as mangrove associate species. So it's rather a fresh look to how I actually look at mangrove associates as well. One thing that we do not often mention, I believe, when we talk about mangroves, we always group them as mangroves, bakau, but when we look at the evolutionary perspective of mangroves, they are actually not one thing. They do not come from one ancestor. All right, they are what we call polyphyletic. They are not monophyletic. Okay, so they actually come from various branches of plant life. Okay, and they achieved similar, whether it's morphology or functional wise, okay, through a process called convergent evolution. Convergent evolution, put in very simple terms, a bat is a mammal, a bird is a bird, but they both converged in terms of functionality to achieve convergent evolution, to fly. All right. So in terms of mangroves, we can very clearly see over here in this plant phylogenetic tree over here, we have the nipa, which I believe many of you know is a very a charismatic mangrove species over here that belongs to a whole different order compared to other kinds of mangrove species as well. So you can see the mangroves, different mangrove genera are actually interspersed in different plant orders over here. You have the monocots, and most of them are the dicots or the eudicots. All right. Then here are the ones that you probably are also very familiar with: Rhizophora, Bruguera, Candilia, Syrops. Okay, these are very common in Malaysian mangroves as well. And currently, in terms of the total, it involves around 18 families and 32 genera. All right. And with all this diversity that we have in the mangrove environment, we can see in certain population patches, especially in significantly large population patches, zonation. All right. So you can see from terrestrial forests towards the sea, you will see different distributions of mangrove genera, okay, group rather generally. But in Malaysia, we also have a lot of small patches of mangroves that do not necessarily follow such a distribution, depending on the topography of the mangrove environment. And as we all know, mangrove ecosystems actually provide various ecosystem services. They provide wood, support the livelihoods, other kinds of services, regulate climate, breeding grounds for fisheries, tourism, water filtration, and coastal protection. However, against the backdrop of all these ecosystem services that are provided for mangroves, human societies have decided to trade that for more food. All right, as in this publication over here, in Southeast Asia, we are a global hotspot for mangrove loss, okay, as well as diversity. And the conversion of forests for aquaculture and rice plantations were the biggest drivers of loss, as well as fragmentation. Okay, later on, we'll talk about what fragmentation means and how it actually endangers populations when it comes to genetic diversity. All right, and unfortunately, Malaysia sits in the square over here with high fragmentation and high loss when it comes to the patterns of fragmentation and loss shown in the study. Unfortunately, it took a huge disaster, a global disaster, to actually remind us of how important it is to have mangrove growing on our coasts, okay, for protection, that actually started a lot of reforestation and rehabilitation efforts and projects around the world. In Malaysia, for example, from 2005 to 2015, we spent around 45 million ringgit to implement this National Mangrove Restoration Project, which at the end planted back, okay, 6 million trees. So think about this. We cleared the mangroves to produce other foods, and because of a disaster, we now need to replant the mangroves with more money, okay? So this is something that we have to ponder, really. So my interest, I'm not really a field biologist. I'm not a strict ecologist. My interest really is in terms of using genetics, okay, to answer certain simple ecological questions. Okay, so basically using genetics as a tool to help these organisms to tell their stories, especially about stories on their populations, because populations are the basic unit of evolutionary change. All right. So we'll go through several slides on the basics in genetics. I believe many of you have learned genetics in high school, probably in university as well, but we'll still go through several terms that I may be using throughout the presentation. All right, so this is something that we see very often. So we have a cell, and then in the cell we have DNA. So where is the DNA? This very simple schematic diagram over here, we have the DNA inside the nucleus. Okay, but this is not exactly accurate. It's just a very simple representation. And in this nucleus, it has clumped together DNA material, okay, into several clumps. And these clumps are called chromosomes. All right. And if you untwine and unclump the chromosomes, you'll actually find strands of DNA comprised of nucleotides. Okay, we call them nucleotides or bases of A, T, G, and C, the ones that you usually hear. All right. Why do I say that this is not exactly an accurate representation? Is because when we look clearly in a cell, all right, we have a plant cell over here and an animal cell over here. We have other kinds of structures, cell structures within the cell, apart from the nucleus as well. All right. And those parts that contain DNA, there are three. So we have the nucleus that people know all the DNA is contained. Okay. Aside from that, we also have the chloroplast in plants, as well as mitochondria in both the animal cell as well as the animal cell as well as the plant cell. All right, that also contains DNA. So right now, you know, there are actually three parts, okay, in a typical cell or typical plant cell that contains DNA. In an animal cell, there are two parts, okay, mitochondria and the nucleus. In the plants, there are mitochondria and nucleus as well as the chloroplast. And whenever we look at the gene, all right, many organisms on Earth, in fact, most organisms on Earth are what we call diploid, meaning to say you have two sets of DNA, one set from the father, the other set from the mother. All right. So two sets added together with the same location and the same function on your chromosome, okay, is called a gene. While the two different components of that gene are called alleles. All right. So why is this term important? Is because evolutionary genetics or population genetics, when we look at genetical change, okay, genetic variation, we actually look at the unit of alleles instead of the unit of genes. All right. So we are looking at the frequency of change. How many of them have how many of the individuals have the red flower allele, for example, and how many of the individuals have white flower allele, for example. All right. Those will then contribute to how this population actually change in terms of evolution. And then next we talk about inheritance. Okay, basically we want to talk about the nucleus, the mitochondria, as well as the chloroplast. Okay, so using an animal cell or animal sexual reproduction as a model over here, during sexual reproduction you have the egg cell, and then in terms of the animal you have the sperm cell, and then when the sperm and the egg actually meets, okay, what really goes into the egg cell is only the head of the sperm. And therefore, in the nucleus, on the nuclear DNA, all right, every person will contain half the genetic material from the father, the other half of the genetic material from the mother. However, what happens to the mitochondria as well as the chloroplast DNA? In most organisms, the mitochondria and chloroplast will be from exclusively the mother. Okay, most, not all, but most. All right. So this gives us a very good tool when it comes to genetic methods. When we look at nuclear DNA, for example, it comprises of genetic information from both the father and the mother. Mitochondrial DNA as well as the chloroplast DNA will then contain information from only the mother. All right. So this one is very useful when we want to tell stories that involve both the parents. And these two are very useful when we want to tell stories or track, okay, the lineage from the maternal line. Okay, so in plants, we usually use chloroplast DNA and nuclear DNA in evolutionary genetic studies. Okay, and that is what we'll be talking about later on. So genes, why are genes important? Why is genetics important? Because genes, as a basic unit of genetics, they affect morphology and function, all right, through what we call the central dogma of molecular biology. So you have your DNA, and then your DNA will be transcribed to RNA. Your RNA will be translated to proteins, and these proteins will be the functional units that will then decide, okay, the function as well as the morphology of the end product. Okay, but of course, genetics is not 100%. It usually or very often interacts with the environment to produce whatever traits, okay, in terms of morphology as well as function that you can actually see in all organisms. So now we enter the subtopic of evolutionary genetics. So evolutionary genetics is basically a study of how genetic variation, okay, leads to evolutionary change. So there is always an evolutionary viewpoint when we do studies on evolutionary genetics. Very often we equate the terms population genetics with evolutionary genetics. Sometimes they can be equated, but sometimes they are rather different. Okay, population genetics, we look at it's very mathematically heavy. So we look at how alleles, we talked about alleles, how alleles actually change within the population and how that actually, drives the evolution of the populations. While evolutionary genetics, we can look at the whole species as a whole. We can look at the species as a population as well, and how allele frequency changes will lead to evolution. Okay, but things like, for example, hybridization studies, adaptation, all these will then fall under evolutionary genetics. All right. So very often when you look at studies on evolutionary genetics, we look at populations firstly, okay, as the basis. Without populations, we can't really look at evolution. All right. And we usually talk about three things. We talk about diversity, so genetic diversity, okay? So the different types of traits that we can find within a population or different subpopulations, all right? We also talk about the topic of connectivity as well as relationship, okay? How connected are these two populations? Do they share genes in terms of gene flow because of migration or because of pollination, for example, okay, long distance pollination? We also talk about relationship. Are they both related? And when we talk about hybrids, yes, then you will need to track who are the parents, all right? And when we involve other kinds of molecular techniques, we also talk about ability, function, all right? So keywords such as adaptation, all right, function, okay, comes into mind, all right? So you have adaptation to cold, adaptation to salinity, for example, all right? This concerns ability. But for this session today, we'll only be talking about these two sections over here. We'll be talking about diversity in terms of genetic diversity, and then we'll also be talking about connectivity and relationship, because when we talk about mangroves, we are looking at different populations and how they are actually connected to each other. And of course, the topic of natural hybridization as well, okay, falls under relationship. I will not feature ability over here because it will take even more time. And I believe this has been addressed by Dr. Alison Wee, one of our previous speakers back in 2019 when it comes to adaptation of mangrove populations. So first, genetic diversity. Genetic diversity is basically variation of genes within a species. So gene diversity, basically, okay? And when we talk about genetic diversity, we can actually talk about different levels of diversity. So when we talk about population biology, for example, our smallest unit is an individual. Then you have a population, which then forms a species. And then after that, you have your community and ecosystems and whatever you have, all right? So in terms of genetic diversity, we will go even deeper. So genetic diversity, we can look at the diversity of genes within even an individual. Remember, I mentioned that in most organisms on Earth, most are diploids, meaning to say they contain two copies of the same genetic material. Okay, one copy from the father, the other copy from the mother. And therefore there is, okay, a diversity within the same individual as well. All right. And when you enlarge it slightly, when you go one level higher, you have diversity within a population. All right. So we look at similarity and differences between individuals. All right. So this is within, this is between individuals. And when we go even one step higher, we can look at diversity between populations. Okay. Meaning to say how much difference, okay, taking an average, how much difference is there between population A and population B. And when we talk about widespread species such as the mangroves, all right, then we are potentially looking at multiple populations regionally or globally, all right, that we can then tell stories about how different or how similar they are. And then from there, we can deduce, okay, whether there is gene flow, whether they are disconnected or they are connected with each other, all right? And this genetic diversity is basically the basis, or they are essential to adaptation, because whatever function that an organism can perform comes from the genes that it actually harbors, all right? So a very simple thought is that if you have very little genetic diversity, if between individuals you don't have much differences, between populations you don't have much differences, whatever that hits, whatever new environmental challenges, okay, that hits a population, and if this population is not able to withstand this change, it can be wiped out. And if this population is not much different from this population, then most probably the fate of this population is also not much different from this population. Okay, so that's a very simple thought of how genetic diversity actually works. And genetic diversity originates from sexual reproduction. So cloning doesn't work, because cloning basically, or vegetative reproduction, basically clones an individual, okay, to become two individuals, but these two individuals essentially share the same or very similar genetic makeup, all right? As well as mutation. And this mutation happens through time, okay? It doesn't mean that today you plant several trees, 10 years down the road, they will mutate into different things. No, okay? It will take thousands and millions of years to actually accumulate these kind of mutations. And therefore, when we talk about genetic diversity, we always talk about preservation of what we have and not about, you know, replenishing or renewing what we have, okay? And finally, number of individuals matter. So this is why fragmentation is so important when it comes to the study of genetic diversity, because fragmentation actually reduces the number of individuals within a population that the individuals within that population is able to exchange genes with. Without these individuals that harbor different types of genes, then you will have very low genetic diversity. And if you have low genetic diversity, it's as good as, you know, marrying within your family for generations, all right? And we all know how that can end up, okay? And just now I mentioned a lot of several times of gene flow. So gene flow is basically movement of individuals, okay? Basically, it's a flow of genes from one place to another or one population to another. And these flow of genes usually will involve migration of individuals. So the individuals, for example, in terms of animal species, individuals, you know, crossing barriers to actually reach the other population. And if they are able to, then they will mate with individuals of that population, and through that mechanism, okay, donate their genes to that population and vice versa, all right? Genetic material can also flow from one population to another population, for example, through pollination, all right? So certain plant species, for example, are pollinated by wind, all right? Then they can potentially cover very great distances. Then you have gene flow at great distances. Otherwise, if you have plant species that are pollinated only by very tiny insects that do not potentially fly very far, and there you have pollination only within a very limited radius, okay? And gene flow can be limited by barriers. So these barriers are what we mentioned earlier. In terms of this diagram over here, it can be physical barrier, geographical barrier, can also be pollinator barrier, Okay, so compare a tiny insect with a bird or a bat. Okay, then we are looking at very different pollination distances or radius. All right. So this gene flow, when we understand this gene flow, then we can understand the concept of population genetic structuring. Okay, so population genetic structuring is basically the organization of genetic variation due to the combined effects of evolutionary processes. So like we mentioned, there can be barriers in terms of geographical reasons, okay, or ecological reasons, distance, temperature. So when we talk about marine species, over here, I've used fish as the model image over here. Fish populations that are established here would probably have a bit of gene flow, but when they come over here, they are limited by land barriers, the presence of land over here that probably do not allow many of them to actually swim over here to mate with the populations over here. And through time, okay, through accumulation of mutations, okay, because populations here definitely cannot just swim very freely across to here to actually mate with the population over here. So through time, we will see a differentiation between populations on the west of the peninsula with the east of the peninsula. All right. Same thing probably will happen across great distances as well. Okay. So over here, we do not have land barriers, but probably, okay, fish populations over here will find it tiring to actually swim all the way here just to mate. All right. There we'll have distance barriers or ecological barriers that can prevent gene flow. All right. And when you have the prevention of gene flow or the limitation of gene flow, what we have now would be what we call population structuring. Okay, this population genetic structuring is a full term. Sometimes we call it population structure, sometimes we call it genetic structure. All right. So basically, it tells us how connected these populations are with other populations. Okay, so these two populations are probably connected, but when we talk about great distances, they are not connected. And then we see a structure over here already. All right. And when we look at the whole of Malaysia, including the peninsula and Borneo, then we will see different subpopulations that otherwise we would not be able to see, because we would think that, oh, fish can swim very great distances. They can swim in water. So as long as there's water, they are able to connect. But it's not always the case. The same thing with mangroves. All right. So mangrove dispersal depends on sea surface currents, for example. Are there barriers to that dispersal or not? Sea surface currents, there are patterns, all right? And of course, across land barriers, they are not able to disperse. And there you have different kinds of ecological or life history traits that will actually affect the dispersal of different species of mangroves, all right? And therefore, whatever planning that we have about mangroves is not mangroves as a whole, but rather individual species with their own specific requirements, okay, as well as life histories. Then with all of these taken into account, okay, population genetic structuring, when given enough time, will actually cause further differentiation. And this differentiation in many species are the basis of the process of speciation, all right? So over here we have a plant, okay, just a plant that occurs on both sides of a mountain ridge, okay, through time, because there is a lack of gene flow, all right, through time they actually accumulate differences in terms of genetic differences through mutation, through other kinds of processes, and in the end they may, okay, it's not necessarily, but they may accumulate differences that do not anymore allow them to mate with each other, even if they have later on meet, okay, each other. They are not able to reproduce anymore, and therefore there we have a perfectly speciated species, okay, two species that are perfectly isolated from each other. All right, so that's the basis of evolution. So from now on, I'll be talking very briefly about what I did for my PhD, so involving genetic diversity, population genetic structure, as well as natural hybridization. And from there, I will try to relate it to other mangrove species. I probably have not read all of the publications up to today, but I've read several, which basically forms my ideas that I'll be sharing over here. And I believe many of you have already heard some of them. So for my PhD work, I actually focused on the Indo-West Pacific Rhizophora species. So for a simple background, there are 3 species in the IWP region. So namely Rhizophora apiculata, Rhizophora mucronata, and Rhizophora stylosa. All right. So Rhizophora species are very charismatic in the sense that they are the most, probably the most well-known ones. Probably people will see them and know they are mangroves, but do not know that they are called Rhizophora. So they have the very typical stilt roots, all right, with the propagules that are long that can float in water. Okay, so forms the impression of many people towards how mangroves actually look like. So this is the distribution over here. Like I mentioned, I only looked at the Indo-West Pacific Rhizophora. On the Atlantic East Pacific, there are other Rhizophora species as well that we do not share with the IWP. All right. So over here, color-coded, we have Rhizophora apiculata that is very much distributed in the Southeast Asian region, extending to a bit on Northern Australia. Then we have some in the Indian continent. Then we have Rhizophora mucronata that has even a wider east-west distribution, and Stylosa that has a wider north-south distribution. Okay, so we can find Rhizophora stylosa up to southern Japan. Okay, I heard even towards Kyushu as well. There are a few, but I have not seen it myself, so I'm not very sure. So during my PhD, that was many years ago. So at that time, we were still doing DNA sequencing. All right, so I did this study using chloroplast DNA as well as nuclear DNA. So just to refresh what we have mentioned earlier on, chloroplast DNA actually tracks the maternal lineage. So basically the seed producer, all right? While nuclear DNA tracks the inheritance of both parents, okay? So Rhizophora species, like we mentioned, look like this, okay? So for those of you who are not familiar, this is Rhizophora. So they have the typical long propagule over here. So basically this is the embryo, this is the fruit. And then when dropped in water, they actually float in the water. Some studies say they float horizontally. I'm not very sure about that, but yeah, so they float in water. They are commonly known as the red mangroves or stilt mangroves because of the sap that they actually produce that can actually be used to dye cloth red, okay? So during my PhD, there were 2 fundamental questions. One is, what is the genetic diversity of Rhizophora species around the Malay Peninsula? Okay, because we had samples from around that region, as well as slightly other regions as well. And what was the population genetic structuring in Rhizophora species around the Malay Peninsula again? Because at that time, there was a suspicion that this Malay Peninsula was a very effective geographical or land barrier towards the dispersal of mangrove species. But then again, they have propagules that actually can float on water and potentially can travel far distances. Okay, so we wanted to know whether that was really true or not. And we also had samples of all 3 species of Rhizophora, so Rhizophora apiculata, mucronata, as well as stylosa. Dotted over here are all the populations that we had samples from, but not necessarily in each population we have all 3 species. Okay, so for example, in southern Japan, we only had Rhizophora stylosa. All right. So I will not go into the values over here, but overall genetic diversity in all 3 species were shown to be much lower than other plant species, terrestrial species. Okay, so thinking about that it's quite interesting and quite surprising, because we thought that with that dispersal pattern of mangroves using sea surface currents, they probably would have accumulated a lot of genetic diversity compared to terrestrial species that would have to depend on wind for their seed dispersal or depend on animal species for their seed dispersal or pollination. All right. So this was quite a surprising thing to find. there were much lower genetic diversity compared to other plant species. And they were especially low within the populations, meaning to say within the populations, there is probably a lot of familial breeding over there. Okay, meaning to say the mother plant will produce the seeds and the embryos will just fall down and grow alongside the mother. Okay, and through time, you will have several generations of the same family within the same population. And that usually is the contributor for low genetic diversity. And we have also discussed about how low genetic diversity is dangerous when it comes to the sustenance of a population, okay, when it comes to adaptation, for example, okay. And when we look at genetic diversity generally in mangrove species in general, so my study was done back in 2013 and 14, published in 2015, and since then there has been a lot of studies on other mangrove species as well when it comes to genetic diversity. There was even I think a review probably back in 2020 or 2019, okay, on the genetic diversity of mangrove species in general, all right, taking a few as models. So basically, there is low genetic diversity found in practically most or all mangrove species that have been studied. When I talk about mangrove species, I'm talking about true mangrove species, yeah, okay. And especially regionally, partly also due to its dispersal ability, okay, because probably near distances, it can very easily disperse. So you can find individuals of the same family, I mean, in terms of the inheritance family, okay, in nearby populations. So unless you sample across, you know, the global distribution of it, then you probably will see a slightly higher genetic diversity in any particular species. All right, and this low genetic diversity is usually a sign of decline. We are not sure, or at least I'm not sure whether or not in mangrove species, whether this low genetic diversity actually holds true as a sign of population decline when it comes to adaptation. Therefore, there are also a lot of studies nowadays that focus on adaptation of mangrove species when it comes to salinity, when it comes to temperature, when it comes to inundation, especially in the face of imminent, what do you call that, climate change, okay, with the rising sea levels, and with the rising sea levels, probably a drop in salinity, and then you have warmer climates, for example. We are not sure how this genetic diversity actually plays a part in mangrove adaptation to all of these factors over here that are definitely changing. All right, so this is probably something, you know, that we can come together to work on. All right, do some greenhouse experiments, for example. All right, and we postulated, or rather, many studies actually supported, okay, these two reasons as the reasons for low genetic diversity. First of all is hostile environment, daily inundation, so daily flooding, salty water, salty substrate. What else? Very hot weather, okay, along the intertidal region. All these are considered very hostile and very strict environmental parameters that a plant population can actually, that a plant population has to endure. All right, so not many plant species are able to adapt to such a situation. And like we mentioned in evolution, we talk about mutation. Okay, whenever there's mutation, the mutation will be tested against natural selection as well. Okay, so all these climatic parameters are factors for the selection on the mutation. And if the mutation does not help the mangroves to advance when it comes to adaptation, then the mutation does not survive because the individual does not survive. All right. So over time, we see, we don't see a lot of diversity. We don't see a lot of genetic diversity. Even when you look at the different species of mangroves, we do not see a lot of species within each mangrove genus as well. Iwp Rhizophora, for example, there are only 3 species, and this is probably the pattern for many true mangrove species, unlike other terrestrial plant species. Okay, so there is significant selection pressure that I believe is causing reduced diversity. There is also issues with pollination and dispersal. We probably have very specialized pollinators that are living in mangrove environments that do not really help them, cross various barriers, ecological barriers or geographical barriers, and so on and so forth. Even for dispersal, all right, when we think a very simple thought that, propagills fall into the water, they can float however far they want to. However, is the sea surface currents actually helping them to achieve that is another issue. Okay, so that's a limitation in terms of the dispersal. So the localization of pollination causes them to actually interbreed within the family. And if the dispersal also limits them within a population, basically over time you have a population that is breeding as a family, okay, crossbreeding with each other only. Then they are, like we mentioned, capable of long distance. They are capable long distance colonizer, but they are very often limited by barriers, sea surface current barriers, or patterns, or geographical barriers. And in mangroves, there is also this central marginal hypothesis that has been talked about quite a lot, but recently it has been rather refuted. All right, this central marginal hypothesis states that in the center or in the middle of the distribution of any particular species, the central part is usually the more diverse, genetically diverse compared to the marginal populations. But we do not see that very, we see that in some mangrove species, but according to this study, it is not consistent because in some mangrove species, it doesn't work that way. All right, so this is the general synthesis for genetic diversity. Then we look at population structure. Remember, I studied 3 species. So we look at first Rhizophora apiculata, one of the most widely studied mangrove species. And in this study, we also had the most samples of Rhizophora apiculata because it's very common. Okay, so when we did phylogenetic reconstruction of the phylogenetic of the populations of Rhizophora apiculata, we can very nicely see a pattern of populations that are grouped into 2 clusters. Okay, and these 2 clusters correspond very nicely with the west of the Peninsula Malaysia, as well as the east of Peninsula Malaysia over here. All right, so this land barrier effect that has been suspected for a very long time actually holds true for Rhizophora apiculata. All right, so this is the barrier effect of the Malay Peninsula, which states that marine species, okay, which mangroves, although they grow on land, but they depend on the seawater for dispersal. All right, so they are not really interconnected with each other when it comes to populations on both sides of the Malay Peninsula. So that one fits very well with the hypothesis. So what about Rhizophora mucronata and stylosa? All right, so for Rhizophora mucronata and stylosa, we had slightly lesser populations, and even lesser for Rhizophora stylosa because it's actually quite difficult to find them. Okay, and even if you find them, you don't find them in large numbers. All right, so for these 2 species, however, unlike Rhizophora apiculata, they do not show a very significant break due to the land barrier effect of the Malay Peninsula. All right, the reasons are out there for us to actually guess. I myself, I'm not very sure. I'm thinking refugia, okay, but there can also be sea surface currents reasons because they actually fruit at slightly different times. All right, slightly. But I'm not sure whether this slightly will actually contribute to such a large effect or not when it comes to the dispersal. Okay, so there you have, when you want to plant Rhizophora apiculata and when you want to plant Rhizophora mucronata or stylosa, you have very different considerations that you have to take into account over that. So population structures of mangrove species in general, not all species are the same. So one message that I want to share over there when it comes to the population structure of Rhizophora, Iwp Rhizophora, is that Rhizophora apiculata, mucronata, and stylosa, they are within the same genus, distributed quite, especially Rhizophora apiculata and mucronata, quite common in Malaysia. However, they have very different, okay, population structuring. Okay, it tells us something. It tells us possibly the ability of dispersal, possibly the origin of dispersal. All right. So for those of you who are not familiar, so basically we have the last glacial maximum. So there was an ice age. All right. The most recently was 20,000 years ago. And during that time, sea levels actually went down, so exposing more land. So that more land was exposed near the Malay Peninsula. Okay, so this whole part over here basically joined up as what we call the Sundaland. You probably have heard of this before. All right, and this Sundaland is basically a very big piece of land that actually barriered the dispersal of species that occurs on both sides of the Sundaland. And with that also, with receding, okay, shoreline, we also see that different mangrove species would probably have to find somewhere to hide. Okay, they do not actively hide, of course, but rather the populations that occur in, you know, places where the seawater was receding would probably die, while those that happen to occur in places that remain as shore, okay, would survive. And those will then serve as the bank, okay, or refugium of that particular species. And depending on luck, if you will, or other kinds of ecological requirements, different species may have found refugia in different regions of the world. All right. And that refugia will then affect their dispersal patterns, okay, across the region nowadays. So 20,000 years in terms of evolutionary time is not very long. Okay. So during that time, they cannot, first of all, they could not accumulate a lot of mutations. And like I mentioned, hostile environment, those mutations might not survive. So in the end, we do not end up with a lot of mutations. All right. And now we have such population structuring patterns. So past refugia, life history traits coupled with oceanic currents. So different times of the year, you have different patterns of oceanic currents. That will also affect the dispersal because your mangrove species will have different fruiting times, all right, fruiting seasons. And depending on when the fruit actually falls down to the water, it will then follow the sea surface currents at that particular time. Okay, that helps with its dispersal. So over here, Duke has reviewed, okay, the dispersal barriers that has been found in mangrove species across the years, all right, to actually include all these regions over here. So that's quite a lot when we look at mangrove species, because different species, even within the same genus, will react very differently, okay, to these kind of barriers. And these barriers can have, whether they are limited by temperature, they are limited by land barriers, and land barriers usually will have to have significant sizes in order to limit the dispersal. Then you have distances, and genetic discontinuity over here would most probably be related to land barriers or other kinds of sea surface currents. So one of it being the Malay Peninsula over here that we have seen causes a difference in terms of population genetic structure of Rhizophora apiculata, mucronata, as well as stylosa. All right, so this is the general pattern in population structure. Then finally, we go into the topic of natural hybridization. So throughout the sampling process, we actually stumbled upon one of the populations in Port Dickson that contain trees that look very different from what we have seen. So actually, I was actually brought there by Dr. Chan Hong Tak. Not sure whether some of you probably know him. All right, so we wanted to know what they actually are. We actually suspected they were hybrids, but we were not sure what hybrids they were, because on that particular, or at that particular location, there were all three species over there. And the hybrids between each of them looked very similar. They were very luscious, okay, big trees that grow very sideways as well. Slightly different from what I've seen in pure stands, okay? So that is where the idea of natural hybridization studies actually came about. All right, so just for your information, there are three species of Iwp Rhizophora again. When we actually identify them, okay, in the field, for me, at least, I look at the leaves as well as the flower buds. All right, so in terms of the flower buds, for apiculata, you will see a very short and stout peduncle supporting usually two flower buds. stylosa, you will see a slender and longer peduncle, quite similar to Rhizophora mucronata, but very often the flower or the inflorescence bunch of mucronata is slightly larger than stylosa. And to assist that, okay, we also looked at the leaf morphology. So Rhizophora apiculata is usually a lanceolate shape. Mucronata is slightly broader, rounder. Stylosa is usually smaller compared to mucronata, and usually the broadest area is slightly towards the top of the leaf. All right. So using those morphological characters, we actually tried to identify species in the field. Usually it is quite easy, but in this particular population, it actually poses certain challenges to us because sometimes we do not know the difference between stylosa and mucronata. And as you can see over here, they're actually quite similar. All right. So in this population in Port Dickson, we found individuals that displayed leaves like that, very often with ridges like these of light color pattern leaves. We are not sure why. And at the same time, they also have flower buds that actually resemble Rhizophora apiculata. So our first hypothesis is that this could probably be, if it's a hybrid between mucronata as well as apiculata. However, the results turn out to be another case where it is actually Rhizophora apiculata and Rhizophora stylosa. Now, at that time, hybrids between Rhizophora apiculata and Rhizophora mucronata has been known. Okay, hybrids between apiculata and stylosa has also been known through a simple observation of co-occurrence of parents. Okay, so you see apiculata and you see mucronata in the same population, and you have one hybrid over there. Okay, so it's a simple postulation over there that this hybrid is a hybrid between apiculata and mucronata. However, on that particular location, we have all three species present in that location, forming one visible hybrid. Okay, so we are not sure which they were. So, like I mentioned, the results show that they are actually hybrids of Rhizophora apiculata and stylosa, and it's a very special population because in that population we have more hybrids than the pure species, than the true species. Okay, and we do not see this situation in other populations, not even in one of the other locations such as we have Bagan Lalang, for example, Bagan Lalang Beach, which also has all three species there, but I haven't seen one individual of hybrids in that population over there. So I myself, I'm not sure why in one location with three species, so many hybrids are there, the other location, no hybrids at all. I'm not sure why, all right? And then it then brings up another question. What about Rhizophora mucronata and Rhizophora stylosa? Because they are morphologically very similar, even if there are hybrids, we probably are not able to, identify them. And like I mentioned, when I was sampling in that location over there, I had difficulties to actually discern between stylosa and mucronata. And probably, who knows, some hybrids are actually amongst them. Okay, so then it brings up two more questions. Do Rhizophora mucronata and stylosa actually form hybrids? If yes, are their hybrids fertile? Okay, meaning to say, are they able to produce, okay, fertile, are they able to produce offspring, and are those offspring fertile or not? So for hybridization study, again, we use chloroplast DNA as well as nuclear DNA that tells stories from both, okay, sides of the parents. For the first hybrid that we saw, that visibly we saw in the population, okay, we determined that stylosa were very often the maternal parents, and Rhizophora apiculata and stylosa were both the parents, okay? And when we look at the population, at that time, there were actually 2 or 3 individuals of apiculata only, while stylosa is interspersed throughout that whole population. And in the middle, there will be a lot of hybrids, okay? And those hybrids were found to be F1 hybrids, okay? So how do we determine F1 hybrids is that, remember we mentioned that when you have an offspring, a child, okay, you have half the DNA material from the mother, the other half from the father. And when you are dealing with two different species as the parents, okay, of course, between the species, they will have consistent differences in terms of their genes, all right? So looking at these genes, if the child consistently have one copy from the father and one copy from the mother at where these two species are different, then we are quite sure that these hybrids are actually F1 hybrids, meaning to say they are the first generation offspring and they do not produce following generations, all right? So this is what we saw. So basically, let's say we have apiculata genes over here. We have, let's say we investigated 4 genes. And then stylosa, we also investigate the same 4 genes. And then at locations where they are consistently different, okay, we see an overlap in terms of the pattern, okay, in the hybrid individuals. Then we are quite sure that they are hybrids. And in this case, they are consistently so in all the individuals. Therefore, they are F1 hybrids, okay? However, when we deal with advanced stage hybrids, imagine you have half the genetic material from father, the other half from the mother. And if this offspring is able to mate with another individual, then you will have reshuffling of the genes in the offspring of that individual. So what we will have end up with is when you investigate multiple genes, then you will have some genes that show you the pattern that we saw earlier on, where the hybrid individual actually share half the gene with one parent, the other half with another parent. But when you look at another gene of the same individual, it shares both genes with one of the parent only. If you see such a situation, it means we probably are looking at advanced stage hybrids, okay? Sometimes people will use the term introgression, where the genes have already introgressed into that individual, all right? So the first visible hybrid that we determined was an F1 hybrid. Now, what about Rhizophora mucronata and Rhizophora stylosa? So when we stumbled upon that population, I went back to look at my data again. And what I found was in locations where there were both Rhizophora mucronata and Rhizophora stylosa present in the same population, certain individuals started to display, okay, patterns of admixture, patterns of mixture in terms of the gene content, okay, between the two species. When we only looked at populations with only one species, either Rhizophora mucronata or either Rhizophora stylosa, they look pure, okay? So these bars, each bar actually represents one individual. And if you see a mixture of 2 colors within the same bar, then you will see a mixture of genetic content, okay, in that particular individual. So mucronata is pure light gray, stylosa is pure dark gray. And in certain populations, for example, this is, I believe it's in Kedah, and we have Bagan Lalang, then we have Port Dickson. I think this was in Jakarta, this was in Bali. We see individuals that have mixed genetic content within some of the individuals, representing that some of these individuals are actually hybrids between mucronata and stylosa, all right? And when we ran another analysis on them, that actually helps to tell us whether these individuals are F1 hybrids or F2 hybrids or back crosses to parent A or back crosses to parent B, all right? We see differing patterns in different populations, showing how dynamic the mating can be when you have both Rhizophora mucronata and Rhizophora stylosa within the same population, all right? So that was the story about my PhD study. And now I would like to just very briefly zoom into the hybrids in Port Dickson, because this is a place that I've been to several times. Sometimes I go to Port Dickson just for fun, and then I just go to that place to actually look at them to see how they've grown. Because this was actually the population that actually started my natural hybridization studies on mangroves, the idea of it. So at that time, we did a brief count of the species composition on that island before knowing that Rhizophora mucronata and stylosa can actually form hybrids, and they form hybrids on this island as well. The visible Rhizophora hybrids, which is the hybrid between apiculata and stylosa, actually constituted almost 70% of the individuals on that island, compared to all the other 3 supposedly pure parental species. All right, and Rhizophora apiculata, I remember very clearly there were only 3 individuals that we saw on that population. All right, and now that we know that Stylosa and Mucronata can actually interbreed, I'm not very sure how many are pure and how many are not anymore. And unfortunately, at that time, I believe it was in 2012 when I visited that place, I saw several plates like this that identified the mangroves on the background as Rhizophora mucronata. Yes, the leaves look very like Mucronata. However, they had the pattern that we saw earlier on. And with the flower buds, we are very sure that it's actually not Mucronata. But there were plates like that around that place. And there were also at that time 1 replanting project called Project Rhizo that were planted in 2011, planting of 2,000 Mucronata seedlings. I believe because of the information that they've received that this is actually Mucronata. And based on one of my unofficial trips over there, I met somebody over there, which actually told me that the seedlings are actually sourced from Kuala Selangor. I'm not sure how far that's true, but this is just what I gather from visits to that place over there. So what I'm worried right now is that the hybrids in this place are very rare. So far, based on publications at least, this place is the only place in the whole world that we know that there is a thriving population, not thriving 2 or 3 individuals, but a thriving population of Rhizophora hybrids. And besides that, we also have hybrids of Mucronata and Stylosa growing on that island or that location. Shows us how, or at least to me, how precious this island or this place is for our further studies. Because right now we do not know why hybrids form in certain locations, but not in other locations when you have the same composition of parental species in multiple locations on Earth. Why are the hybrids only limited in certain locations? All right, and with the I would call this misplanting of species that I believe should not be there or should not be there in such large numbers. Now, I think I last went there before MCO. The trees were already flowering and fruiting already, the planted ones, and they look quite significantly different from morphologically from the Mucronata that we have in that location as well. So that actually worries me because it actually then changes the dynamics of when it comes to reproduction, okay? Whether or not the hybrids will assimilate themselves with the Mucronata over here, and then you will just make everyone a Mucronata when it comes to Stylosa and Mucronata. And the rarity of Stylosa actually worries me also, because then I'm not sure whether this planted Mucronata population will then replace the Stylosa population that is in that location. That is also contributing to the Rhizophora hybrids that we actually see on that in that location. All right. So when it comes to hybridization, like Audrey mentioned in the beginning, there isn't a lot of studies. Okay, the latest one I believe was a review by Raghavan, which I also contributed information when it comes to Rhizophora, Iwp Rhizophora, that was published in 2017, that reviewed hybridization in 7 genera. Okay, out of, like we mentioned, 30 to 32 genera of true mangrove species worldwide. All right, and I just very simply put out a table summarizing what we have in IWP and IWP, not necessarily in Malaysia. So Rhizophora, we have 3 IWP species forming. Right now, we know that there is 3 hybrids, and then you can see that the number of species, like we mentioned earlier on, is not many in each genus, and all of them consistently have hybrids. So all these are very major true mangrove genera. And based on the year of discovery, we can see that it is not all that were discovered very early in the years. Many of the early discoveries were from, I believe, Tomlinson and Duke as well, okay, back in Australia. And then the later ones would be individuals that were probably identified through genetics, okay, through molecular studies. So I feel that those that has many species, such as Avicennia, we probably have many hybrids that are still undiscovered and would be interesting to actually look at them. Rhizophora, I believe they are discovered because they are one of the most widely studied species, Sonoratia as well. All right, so I think we have a lot of work still left in this area. So how does genetic diversity, population structure, and the discovery of natural hybrids actually inform or contribute to conservation and management? All right, so right now, a lot of the conservation and management work, it's probably about conserving what we have and planting more trees to rehabilitate or reforest certain mangrove areas. However, I'm not sure, because I'm not involved in these planting efforts, I'm not sure whether or not, when it comes to replanting, the stock where the seedlings come from is actually taken into consideration. Maybe this is something that we can discuss later on. And I would like to urge for these kind of efforts to actually take into account genetic diversity when you decide on what populations to prioritize, what you want to save and what you want to let go, because it looks degrading, that the population is degrading, but probably that population contains a high diversity that you probably want to save, you know, in consideration of the long term when it comes to adaptation to climate change, for example. And for such efforts to actually take into account population genetic structure during reforestation, Kuala Selangor, taking Rhizophora mucronata stylosa seedlings from Kuala Selangor, replanting them in Port Dickson, I don't see a major problem in that. But the fact that that population in Port Dickson is very special, I actually am very unsure and a bit worried about the future of that population over there when it comes to the survivability of the hybrids that are rare and worthy of more studies. But say if you were to take seedlings from Kuala Selangor and replant them in the eastern peninsula coast, okay, that would worry me a lot, okay, because then basically you're assimilating the genetic content of the west coast with the east coast in the long run, because they will reproduce, right? They will mate and reproduce. Whether or not the individuals that are adapted to the conditions in the east coast, when mated with the individuals that are adapted to the west coast, will actually produce offspring that can adapt to the conditions in the east coast, or do they reduce their ability by half? Okay, so that is something we are not sure, and therefore it is quite worrisome if we were to do that. And to decide on the status of the protection of natural hybrids, how are we going to look at natural hybrids? Are we just going to look at them as hybrids? If they are gone, then they are gone. Or are we going to classify them as rare taxa that requires our protection and further appreciation? All right. So for example, Bruguiera heinesi over here, that was described as a species initially, was then later shown to be actually a hybrid between two other Bruguiera species. But this heinesi taxon has been listed in IUCN Red List as a critically endangered species because they don't occur in high numbers. Are we also going to list, for example, Rhizophora X lamarckii as one of the endangered taxon? All right. And finally, the question of to plant or not to plant, I think it really depends. So if you want to rehabilitate, if you are able to, I believe sourcing from that original local population for seedlings is very important to actually maintain the local adaptation of that population to that condition over there, and not importing it from some other locations to a very far away location. So careful cataloging and planning of mangrove areas is very important, because you need to know what is there. Are there hybrids over there? Are they forming hybrids? Do you want to protect the hybrids? If you want to protect the hybrids, by increasing the number of certain species, will it affect the survival of the hybrids or not? Okay. And like I mentioned, more ideal to conserve and manage existing mangrove populations than to create, okay, new mangrove populations. And finally, understand ecological requirements when deciding on species to be planted. Are the substrate actually suitable? Is the salinity over there actually suitable? Are you planting a certain species that are meant to be, slightly towards terrestrial region at the seaward region? Okay, all these has to be taken into account. So I think I will stop here. Thank you very much for listening. I'm really, really humbled by the attendance of colleagues and VIPs in the research arena. Finally, I just have this to say: saving a species is one thing, saving a species because of lack of information is another. Thank you very much.
Thank you very much, Wailun, for this eye-opening lecture. So much knowledge we've learned. I think you may want to keep the slides on for a while. Perhaps
okay.
some questions will come,
Sure.
we want to refer to the slides. Yeah.
Mm-hmm.
But yes, I think I truly agree with you towards the end where you touch on the importance and that we have to be wary, right, on this knowledge on these important characters and properties of mangrove, in which you've already correctly highlighted that everyone is now eager to assist in restoring mangrove. But are we doing it right? Yeah? Basically, we know of the basic requirement for mangrove to grow, the physical characteristics and whatnot, but very lack of information actually consult or local communities, authorities on this matter, on this perspective. And yes, if we don't scream and if we don't disseminate this knowledge, particularly in your case study in Port Dickson, yeah, people will simply plant, yeah, as to fulfill their needs or their eagerness of replanting and restoring, but that affects a lot of other things. Yeah? I like this ending quote where you say saving a species is one thing. Yes, we want to protect mangrove, but are we doing it right? Yeah, with is it are there enough sufficient information and knowledge to decide on that? Yeah. And as many of us agree, actually, we would always push or suggest for, like protecting the area and let nature regeneration happens. And yeah, perhaps that there must be ways for us to do it better. Yeah. So okay, that's just a comment. But then let us go through the chat, if there are some relevant question, or if there's anyone who want to ask a question directly, you may do so. You may raise your hand or just turn on your microphone and your camera so we can.
Dr. Fong here.
Hey,
Hi.
thank you, Fong.
You have to go without the camera. Not really grooming well this morning. There's one interesting question. Yeah, when Wailun, you touch on why the low genetic diversity in Rhizophora. One of my students, she did some propagule dispersal at the coast in the southern part of Penang Island. Now, what she found out, as compared to Avicennia and Rhizophora, she only compared two species. Yeah, we found that Rhizophora, because of the size, you see, and also when it falls, right, the propagule get tangled near the mother trees, as compared to Avicennia. Avicennia, the seed, is it's round, way smaller. Yeah, it get dispersed very far. So that could explain one of the reasons why you have this, Peninsular Malaysia as the land burial for Rhizophora distributions, etc. And another thing is it's because of the surface current. We are located, especially in the northern part of Peninsular Malaysia. We are above the equatorial, though not much, but we have this Coriolis effect. It's due to the tilting of the Earth, on its axis, you see. So overall, all those we find a similar patterns in seed dispersal in Penang as well. Eventually, all the seed dispersed, right, will, if it get washed away from the mother tree and it get carried by those long shore currents, they will go to the north if we are at the northern hemispheres of the Earth. So that is a Coriolis effect. I'm not sure whether that ties in with your findings, saying that Rhizophoras, they have this land burial. Peninsula Malaysia is a land burial of that. And second observation, I do collect some Rhizophora mucronata and stylosa trees, and because I study on the pollen as well, yeah. So when I was in Bagan Lalang about 4 or 5 years ago, yeah, and then I found out, though Wai Lun said you never observe any hybrids between these two species, stylosa and mucronata, but I observed some younger, trees. They're about less than 10 years old, and they're way out further to the coast. And it confused me, you see, because from the leaves and also from the flowers, it looks as if, they're stylosa. But, the leaf is way bigger than stylosa. So I thought those were hybrids, you see. So when I brought back to the lab and then I opened up and I studied the style of the flowers, I found out the stylosa is actually very distinctive between the stylosa styles and the mucronata one. Mucronata is quite the opposite of being a big leaf guy, but the styles of the flower is the shorter one. Yeah, okay. So I found out there's actually no hybrids. But if you rely on just the size of the leaves to differentiate, to physically different, morphologically differentiate these two species is quite difficult. Yeah, I'm not sure whether you have done other comparison to look at the other parts of the flowers, or maybe pollens, or maybe the styles to differentiate between stylosa and mucronata, Dr. Wai Lun?
Okay. Thank you very much, Dr. Fong. I believe there are two questions over here. One is regarding the land barrier effect of Malay Peninsula, relating to also the size of the propagules and also the constitution of mucronata and stylosa in Bagan Lalang. So am I right? Then I'll address.
Yes.
Yeah. So I'll address the Malay Peninsula question first. I'm not very well versed with the sea surface currents, but what we can see over here is a very clear distinction of populations on the west as well as the east of the Malay Peninsula that contrasts with mucronata and stylosa. So when we look at just apiculata and mucronata, apiculata has actually a shorter propagule compared to mucronata, and mucronata's propagule is heavier, so it can grow up to 60 cm, and apiculata is about 30 cm. So you have a double size over there when it comes to mucronata. So I am not very sure whether that actually affects how far they actually float. But if you think about physics, the heavier it is, the more flotation force there is, and then probably they can float longer. But then again, it also concerns sea surface currents that can bring them anywhere. And when we look at mangrove populations, they usually occur in patches. They don't occur as a continuous strip of mangrove. And therefore, whether or not they can succeed in reaching another patch is another issue as well. And yes, we also see a lot, when we go sampling, we also see a lot of propagules basically stuck amongst the roots of the mother tree. So that actually encourages a familial breeding. So we have seen a lot of inbreeding over there. That is one of the major reasons for low genetic diversity. All right. So as with whether or not oceanic currents actually can contribute to, I believe they can, because also they fruit during different seasons, different times of the year as well. And I think there would be alternating currents at different times of the year that will contribute to their seed dispersal and therefore the patterns of the population structuring as well. We'll go to the question about bagan lalang. So I'll show you the results of bagan lalang. So this BLS that we see over here is actually bagan lalang. So there, amongst the many individuals that I actually sampled over there, I believe there were probably around 20 lah. Okay, only 2 individuals actually displayed a hybridized pattern when it comes to genetics. I also know the few trees that you actually saw very towards the sea. I thought they were quite special, very nice when you take pictures. But I feel they are probably plasticity, because they will then have to adapt to the conditions over there. If you take a closer look, you can actually see that their roots actually grow in one. It's biased towards one direction, and it's actually quite special to actually look at. And the trees are usually quite low because probably they need to adapt to that flooding and the very loose substrate that they actually grow in. But those individuals over there, I sampled quite a lot of them, and I believe they belong over here. They are quite pure in terms of stylosa when it comes to stylosa, aside from 2 individuals that probably have contributions of genes or genetic material from Mucronata. So yes, and yeah, you also asked about morphological studies, right? Whether or not I've tried to characterize. So I think it was last year, 2020, I actually published a paper in Malaysian Nature Journal. I can share the paper later on in the chat group. I forgot what was the results, but several students actually measured some characteristics of the inflorescence. So there was some significant differences between these 2 species. But anyway, I don't really remember the details of it anymore. But I'll share the paper and let's see what we can do about it, because I'm really very interested in doing this. Like you mentioned, stylosa, the name stylosa actually implies that there has something to do with the style. So basically, stylosa has a longer style, usually around, I believe it was 0.5 cm, so 5 mm of style in stylosa species when you break open the bud. While Mucronata, it has a shorter one, so probably around 1 or 2 mm. But then again, these quantitative traits, sometimes it is quite difficult to really segregate them apart, especially when you're dealing with hybrids, and more so hybrids that can form advanced hybrids. Okay, so then you will have all kinds of intermediate structures that are forming along that whole spectrum of differences between the 2 pure species. When I mentioned that I did not find hybrids in Bagalala, I meant I did not find the visible hybrids, which are the hybrids between Apiculata and Stylosa over there in Bagalala. Okay, so that's my answer. I'm not sure whether that answers your questions or not.
Yes, you did. Thank you, Wailun. Yeah.
Thank you very much for the question.
We were looking at dispersal patterns and diversities of Avicennia. What about that? I mean, since we found out the seed of the Avicennias, actually they disappeared, within 5 days. Mm-hmm. Once it dropped from the mother trees, within 5 days they will totally wash away from the coast. So, and that partially explain, why Avicennia, especially Marina, is the widest spread in terms of, distributions and all that worldwide.
Yes.
Any chances of you doing similar project on another species, Avicennia?
I think there has been other population genetic studies on Avicennia out there. I did not really go and read the paper very clearly, but I can try to go and take a read. But there are studies that shown, different sizes of propagules or different propagules from different species that can actually survive in water for different lengths of time. I'm not sure whether that will also affect or not, because even if they can disperse to long distances, they may not survive the journey. They may be too long submerged in water and therefore cannot survive anymore. So I think these are very fundamental biological aspects of things that we can actually work with, for example, with more junior students, final year project students, to actually come about like different species, how long do their propagules, survive in submerged, in saline water, for example. I think those are very interesting questions that we can actually look at.
Yeah. We just a short input. There's actually a student who did just that. To look at the survival or the buoyancy of the different species of propagules. They found out for Rhizophora apiculata, it can actually stay afloat for 2 months.
2 months.
After 2 months, then it'll start to sink, you see. But for Avicennia, it can go beyond that. Yeah. There are studies on this. Yeah.
There is also a very early study, I think also on Rhizophora, that mentioned that after 21 days, the propagule basically don't survive, although they are still floating. So survivability is also something. Survivability is also something that we can look into. And also, when you talk about pollination, I think this is something very interesting that I think we have discussed before also, probably, on what can be studied when it comes to pollination. So when you study pollens, for example, I'm not sure whether you can check on whether the pollens are actually viable, for example. So for example, in certain hybrids in mangroves, people have looked at the pollen on whether they are viable or not. And they found that in hybrids, the viability is actually very low. So I'm not sure how that can be done, but maybe this is something that you want to look into.
Yeah, that sounds interesting. But in terms of morphology, right, the 3 species of Rhizophora doesn't produce a lot of difference when it comes to pollen structure. Yeah.
Pollen structure. So it
but viability, it could be the next things to look into. Yeah.
Yeah. So it actually lays a basis for them to actually hybridize with each other.
I believe so too. Yeah, I believe so.
Yeah. And I was actually quite interested in doing pollinator studies, but the idea is still very premature as to why 2 different locations with the same species composition, one produces a lot of hybrids, the other one doesn't produce hybrids. Of course, there can be other environmental conditions as well, but could pollinator composition actually play a role in terms of the possibility of them hybridizing or not?
Yeah, that's a big possibility. So it's time to fix some cameras onto the trees, Wai Lun.
Yeah. Probably we can ask help from Audrey for that. Audrey,
Yeah, obviously, thanks Dr. Fung for the questions and comments. Yeah, I think obviously, Wai Lun, there are a lot of opportunities, a lot of subtopics to be studied, and of course, to do that, we need some pure stands, we need some pristine, undisturbed area to properly understand hybridization, speciation within mangrove. Yeah? So I hope, some reserve area would be, perhaps this is a whisper to the managers, to the forestry department, or maybe the fisheries department, to consider this. Reserving some area, some pristine area, by avoiding maybe interruption from planting, from other activities, to allow for a specific studies such as this. So maybe one step that we can take at the moment, Wai Lun, to properly demarcate or identify this specific area that you've already spotted at the moment. So we can suggest this to the managers to consider, and then to maybe provide some allowance for us to focus it on this particular study. Because I like that idea. I'm pretty sure, I think Stephanie just left, but she's very interested in pollination from my understanding. So it could be wind, right, for pollen, most of the time maybe for mangrove, but of course we also know Brugerias, Soneratias, I think Soneraceous, it's pollinated by bats, for example. So these are some of the unknowns still within mangrove also. So there are a lot of opportunities here. So this is an out loud request to everyone, especially the younger generation of scientists to come in and join and contribute to this research. I have one specific question, but I see Maiza raising her hand. So let's hear from Maiza.
Hi, Dr. Audrey. Hi, Dr. Wee Leong.
Hi.
This is Dr. Maiza from University of Malaysia, Terengganu. Thank you so much. I just want to say a very quick comment. Thank you so much for your very good sharing. I don't have a background in genetics, but it makes your lecture today just taught me a lot about genetic, and then I can easily understand from the beginning, or from the initial ideas of what genetic is. So maybe I just wanted to highlight this to Dr. Audrey as well. The main point that I get take-home message today, very important take-home message today, is that it's not because mangrove can be replanted that we can just demolish an area for us to use that area and then replant again. But the most important thing is to preservation of the genetic pool. So I think probably if Dr. Audrey can also not only teach us the scientists, but also make this message delivered to the management of the, those pihak yang terlibat lah, okay? So I really support the effort of Dr. Audrey always writing on the newspaper and so on. So I think this very important and easy to understand take-home message should be well delivered to the management of those people who involved. So well done. Thank you so much.
Thank you, Maiza.
Thank you very much.
I think I truly need input from Wai Lun for that, truly. Yeah. But yes, not just hybrids, not just genetic diversity within the plants. And if you remember this recent paper by Joe Lee and his team in Hong Kong studying faunal, there is very low redundancy of roles and function played by faunal organism in the mangrove, meaning meaning they are dispersed throughout the habitat, and each faunal organism play different role within the system. So if we simply bulldoze or simply clear an area, we could have missed that very particular important faunal species that play the role for the whole habitat. So this is another thing. Of course, we cannot see, from our, naked eye, we cannot see that process, that function playing, right? So, but then through science, through this explanation or scientific description, we know of that fact. So there are more things to be understood, to be, make known by for managers and forest managers in particular, and that also, of course, leads to protection of that area. So good. Thank you, Maiza, for highlighting that. And yes, please, Wai Lun, if you have idea, please jot down. And if you feel like you need assistance, to collaborate or to get inputs, and just say it out loud so we can figure out some of our connections, and then try to push for stronger impact, like getting grants for the study, getting areas protected. Yeah.
If I may add,
yes,
because I think I can see you can be our mentor of how to, you always manage to write things in a layman terms, and then probably with the main message in this presentation of Dr. Wee Leong, I think if you can help us to simplify further and then put it into a language that many people can understand on the newspapers, I think that will be very asking for another research grant is one thing, but I think it has a huge and very important message in this presentation, and it should be probably for one presentation we convert it into one publication, or for the public publication. I think is very good idea as well.
Good. Sounds good. Yes, yes, we will take note of that, and thanks for the support and thanks for the motivation as well. And then messages to the public, yes, that could help. And then, but then of course it needs to be supported by more information, more stronger data. But then now we have actually solid information already, particularly for Bagan Lalang, for Port Dickson. Okay, so now when we speak about human or anthropogenic impact on species genetic diversity, we cannot ignore also some other impact by humans, as particularly river pollution, pollutants coming from industry, and lately, I mean recently we had this discussion on oil spill as well, and it's affecting Tanjung Tuan in Port Dickson, Wailun. So I'm pretty sure it was your site actually, I think. So I don't know, was it in Tanjung Tuan, near Tanjung Tuan, right? The plot.
It's actually very near. But
yeah,
Tanjung Tuan, when it comes to mangrove species diversity, I think there's not much over there.
Yeah,
there's one huge patch of Rhizophora apiculata pure stand.
Correct. Yeah. And they are badly affected, and from that oil spill, and not there are like it's a mixed patch of Zoneraceae and Rhizophora apiculata actually. So apiculata was affected by the oil, but not the Zoneraceae. So this is interesting, very interesting thing also to understand. And yes, surely there is more to be explored, and yes, we will take note of all the suggestions. There are some input actually in the comment in the chat section. For example, Stephanie earlier in the talk, she actually highlighted like make sure also for us to emphasize the role played by fauna and flora, and in addition to the value of fisheries. I think we all realize that, we all know, understand that, and perhaps more knowledge on other services played by other organisms or other creatures within the habitat is of course important. And I always try to also push the connection with abiotic or physical parameters within the habitat and ecosystem, because that leads to connectivity, habitat connectivity, meaning mangrove and seagrass, for example, mangrove and coral reef. So there are beautiful things, awesome things to be explored within that network as well. And I think we surely need to know, to understand on how it affects genetic relationship as well. So that one, and then I think the rest, everyone is thanking you, Wailun, for a very awesome presentation. In fact, they say this is a crash course for genetic 101.
Thank you very much for appreciating.
Okay, there's one from John Yong. For replanting in Malaysia, is there a consideration for population genetics in terms of where the propagules are sourced? Yeah, this was what Wailun raised at the end of the lecture just now. I mean, from my observation, from my understanding, basically no. From what I understand, they simply purchase it, especially in this recent race. It is not obvious in Malaysia that we target to plant like millions or hundreds of thousands of species, but we hear about this from any other areas in the world, throughout the world, that they are racing to plant as many seedlings as possible. But the question is, where did they source this? I mean, like, for example, 1 million seedlings, right, in 1 year, for example. And where can they source like 1 million seedlings, right? I mean, if we have like limited area, particularly for smaller countries with smaller areas of mangrove, so it is impossible. Surely there will be disturbance. Of course, we have a nice intention, but then the methods or the approach may not be suitable. So there you go. So I think, yes, it is a good point for us to raise, and based on this lecture and discussion today, I think it is a need to consider. And we did advise actually based on, if you remember, I did highlight about Allison's talk in 2019. In fact, prior to that also, we did raise a concern about transplanting transplanting it or purchasing a seedling from different localities. So, and then there's also another paper. I think I can't remember her name, Azman. Published from Malaysia, from genetic diversity, and speaking about Peninsula Malaysia as the species barrier. Emilia Azman, I remember that paper. And then of course paper by Alison Wee. So maybe we can create a subgroup on this Wailun through MyMangrove, where we can push this idea forward. And I see Prof. John Young. Hi, good morning. From where you are, we are already 1 p.m. here in Malaysia.
Can I just make a quick comment? So Wailun, thanks for marvelous thing, and it's good to meet you face to face. So
yeah,
just to share with everyone the issues that we face in Singapore. So I got into trouble when I said certain things that Wailun has said to the authorities. So just to be honest, so we had the same issue. I don't have very strong background in genetics like Wailun, but the issue that we faced was, like, for example, our sonar ratio comes from Thailand for replanting in Singapore. So the reason for them not to consider all this is simply the cost of propagules or saplings. And in Singapore, we have Pulau Semakau, which is a very big island that we actually have to do a lot of replanting. I raised the issue that the propagules came from very far away islands like Lingga and even up to Java. And it is simply because of the cost of propagule. So the consideration at the moment is still cost of materials, and they do not regard the population genetics. So I just show you one little thing that I managed to gather, and it's actually messed up genetics. But when I brought this up, I got into trouble with the authorities, So, Singapore is very regulated. I'm very open, and that's why I'm in Sweden now, a more neutral country. So I'm not a population genetic, but based on all the inputs from the late Professor Ong from USM, from Nom Duke and all that, I've sort of told the authorities not to go 50 to 100 kilometers. So this is 20 years ago. So that's why I say, Wailun, your data is very important. So Malaysia should not make the same mistake. So the Singapore experience is that because of the horticulture trade and because of quotations, a lot of our Bulgarias and sonar ratio comes from Thailand. And this goes into our park. And now Daniel Fries is handling all this. Okay, but I don't think he has that background. So it's good that if somebody is here, please tell him that. So Wailun, your contribution is very important. Okay,
thank you
but
very
the worry
much.
now in Singapore is that all our you could see our lemacry will be cross-fertilized by the ones from Java. So this is based on your thing is scary. Okay, so our things is coming from Java because it's cheaper. Indonesian rupiah is very small. Then for Bulgarians and Sonoratia, it's all coming from Thailand because of quotation system. So this is why I say it will be very messy in Singapore. So Singapore is actually very far ahead with the replanting. So Wailun, thanks for all your introgression data.
Thank you for the appreciation.
Very important. Thank you for the input, John.
Yeah, and so don't follow the Singapore way and based on quotation. And the East and West is very important. That means staff at Terengganu should not be brought to the west coast of Malaysia and all that.
Surely take note of that. We need to
highlight this. The only thing is, Wailun, you may have to suggest a distance in a recommendation. That means, like, for example, things at Penang where Suyok is should not go down to like Negeri Sembilan, that sort of thing. Yes. So will you eventually come up with a guide? Based on your results?
I haven't really thought of that, but that's actually a good suggestion. But when it comes to genetics experiments, that probably will swallow up some cost and time as well. I'm not sure how we can actually do this.
But that's a very nice suggestion. In fact, actually, I mean, considering our, large patches that we have, John and Wailun, for example, like Perak alone, right? In Matang, it sure can cover like just Perak, right? And the same goes for other states. Actually, we don't actually need to cross state border to get if we need to do planting anyway, right? So, but then maybe the nurseries or operators, right? That could be the limitation factor. But then but then of course if we have correct, right?
Yeah, Audrey. Now to John, hi. To add on to that, right? Fong here from Penang. I've done a lot of replanting project in Penang, basically because Penang has many international company like Motorola, Intel. They always provide funds for replanting. Now there's this particular site where they have all these fireflies, Nibung Tebal. And it's a beautiful patch, of Cassularis, the Sonoratia cassularis, it's Berembang. So we observe that the natural regeneration is really poor because the fishermen boats, every time they go in and out, they rempit sikit lah, they speed, and then all this currents has washed away all the regeneration, all the seedlings cannot survive. So we have to step in and do some intervention. But it's really, really hard to source for seedlings. We search for seedlings, all the way from Perlis to Perak. And, it's like no nursery want to keep all these seedlings, is it? The only available ones are usually apiculata, or sometimes you get a little bit of mucronata. So, and we also try Avicennias. We even give advance notice like 6 months ahead to the nursery, ask them to prepare, give them quotation, give them money first, provide them with polybags and all that. They still refuse to have even Avicennia marina. Because of the roots. You see, Avicennia is a kind of like a pioneer. The roots will actually come out from the polybags and grow wild, spreading and tangle the neighboring bags and all that. So in terms of theory, of course, it's ideal to have localized nursery to avoid genetic contamination when we do replanting. But in reality, it is really hard to achieve. I mean, this is just a warning ahead. Thanks, John.
Correct. I mean that goes to the discussion of limitation in supplies by nurseries, and of course experience as well. We don't have that many experienced nursery operators. So I mean, but I'm still worried, like if there are requests, like for example 5,000, 10,000 of seedlings, and where could they source this, right? And then I mean, of course there are limitations as well, because many quite significant area of mangrove in Malaysia are in forest reserve, right? So they cannot simply enter and source and pluck or pick from the ground just like that, right? So there must be, I think, I hope that at some point we will have some guidelines of operating a nursery. It is a good intention by local communities. Yes, we support that, of course, for their income and whatnot. But it is good also to have like a wider public or communities that have this scientific background or scientific understanding to appreciate these activities better. So I think I think we had like good discussion. I don't realize it's actually already 1 when I saw John Yong. I look at my clock there. 1:05. So good things, like run. We don't realize times are flying, right? Time is flying. So, but then, okay, if everything, if everyone's okay, I have this one final question. I hope to close this discussion. Lately, actually, not lately. In fact, for the past 15 years already, it happens in Moreton Bay. It happens in. I saw this in North Queensland, and I see it in Malaysia as well. Albino. So what is there any effect of, is it are we like contributing to more creation or formation of albino seedlings, Wai Lun, and how and what are the implications, or maybe how for us to avoid this issue from exploding? If you can,
When I was sampling around Southeast Asia, I believe it was in Thailand, but I forgot which location already. When I was walking along the boardwalk, I actually saw certain individuals that produce propagules with very light color, probably a bit yellowish. I'm not sure whether that's what you call albino or not.
I saw reddish in
reddish. I saw a bit yellowish, like it's not really green like usual. So at that time, my supervisor actually said that it's probably an albino. And this albino situation apparently in plants is an effect of inbreeding, meaning to say familial breeding. So probably you have siblings breeding with each other, and then after that, generations and generations regenerating in the particular same location. And then after some time, the alleles for the genes actually are the same. Then that is the basis for albino to actually happen. That's actually one of the telltale signs of the low genetic diversity that we can actually find in a particular population.
So it's not really negative implication to nature rejuvenation or nature regeneration of mangrove. Yeah, I assume.
I think they are quite strong, but in terms of the inbreeding situation and the lower genetic diversity situation will then affect their ability to adapt to whatever change that will happen, climate change and environmental change, anthropogenic change, whatever have you. So I think adaptation studies are important, like what probably Alison was doing and is doing at the moment.
Sounds good. And thanks, John. John just shared another article there in our chat box, so you guys can check it out. And of course, I think, yeah, we have taken a lot of time actually. So thank you, Wai Lun. If you can stop share so we can invite the rest to, turn on their cameras so we can have a quick photo session. But while doing that, I think as a summary, I think for sure, this is obvious. This topic needs extra or additional work, and it needs a more knowledge development, more collaboration in research, and more shout out to the managers, to the government, to the public on the importance of this knowledge and information, particularly affecting relating to our effort in restoring mangroves. Yeah? And of course, we already had issue on planting techniques, yeah? on choosing the right site, choosing the right species, but now we should also scope into genetic diversity, because we don't want to create more issues in the future. Yeah. So of course we can actually lay out the implications, the negative and positive implication from restoration work. So these could be put in the negative section. But of course, we want to try to minimize that, right? And all in all, yeah, good intention must be also supported by good science and good efforts by communities. So I think we learned a lot today from you, Wai Lun. Thank you very much. And I'm pretty sure,
thank you, everyone.
Yeah, I'm pretty sure from speaking for many of us today, we thank you very much again. So hopefully, the traditional MyMangrove monthly discourse in 2019 was we visit the speaker and take a look at their labs, take a look at their the campus, university, or offices, right? We cannot do that at the moment, but hopefully we can have the opportunity soon to visit you at Xiamen University, and perhaps we can start discussing maybe the sharing of resources, sharing of labs, because I myself I don't have high tech lab perhaps like what you have in Xiamen, so we can start discussing about sharing of resources. We can go to UMT to visit Maiza soon. Yeah, Maiza will be our speaker in April. Next one, yeah, next. Oh, by the way, yeah, do we have everyone opening their camera, or I only see like 8 or 7? Yeah. Okay, so another 20 seconds I give for you to turn on. And next we will have Dr. Chi Su Yin from USM. So Su Yin recently published a super awesome paper on mangrove as nature-based solution. So we might hear about the topic, but I still gave her another 2 to 3 weeks to decide on her topic specifically, and her to provide information for the talk. So stay tuned through MyMangrove Facebook group and also our Twitter for information. But I will blast the email and WhatsApp anyway to all of you. Yeah. So okay, so let's have the group picture now. Hang on. Okay, so 1, 2, 3, 3, 2, 1. Hang on. Okay. San Muna Renda. All right. So thank you very much again to everyone, and Wailun, especially as a speaker, and everyone. John, I don't know, it's I think 6 a.m. in your in Sweden.
Yeah, it's 6 a.m.
6
I woke up
a.m.,
at
right.
3. Yeah. So
wow.
Wailun, you must be special. Must wake up for you.
Thank you very much for supporting.
Yeah.
Thank you very much for the support, and everyone. I see Azmil also from Sabah, and Tuan Sulaiman, Farani, Farah, Izwandi, thank you for joining. Puas Linda also there. All right, so and everyone, of course, and thank you for the support for MyMangrove, and of course, this is the platform, as I mentioned earlier at the start of the talk. This is the platform for us to discuss and for us to learn a lot from the speakers and get to know mangrove scientists, mangrove researchers, mangrove managers, and mangrove fans out there. Yeah, we have participation from local communities also from time to time. So again, with that, on behalf of the Malaysian Mangrove Research Alliance and Network, I would like to thank all of you and stay in touch, stay safe, and keep mangroving.
Thank
Happy
you.
mangroving. Thank you.
Thank you, Audrey. Thank you, Wailun. Bye, John.
Thank you very much,
Thank you,
everyone.
Doc. Bye.
Bye.
Okay, the recording has stopped. So thank you very much to everyone. Yeah.